I hereby open this academic ceremony in which Ms. Francesca Giacomini will defend her academic thesis, Making Attachments, Advancing Enthesis Regeneration Through Miniaturized Models. I welcome you and wish you lots of success together with your parnames. And I also welcome your supervisors, Professor Truckenmüller, Professor Habibovic, and Dr. Kieselbrecht, and of course the members of the opposition, whom you will meet later, and a warmly welcome also to the audience here present and online. But before we start the opposition, we give you the opportunity to explain to your family and friends what you have presented in your thesis.
Thank you. Dear Prorector, dear member of the Defense Committee, dear colleagues, family, and friends, in the next 15 minutes, I will give you an overview of my projects and main results. And I would like to start by introducing body movement, which is a fundamental aspect of life, allowing us to interact with the environment, perform daily tasks, and engage in physical activities. Whether we are walking, running, or lifting an object, our body relies on a complex biomechanical system to generate forces and to transmit efficiently. Indeed, behind every movement, there is a highly coordinated action between muscles, bones, and joints, which form the musculoskeletal system. In the musculoskeletal system, muscles generate forces, bone provides stability, and joints facilitate motion. In particular, we are interested in tendons and ligaments. Tendons connect muscles to bone. They attach to muscles through the myotendinous junction and to bone through the enthesis. When muscles contract, the forces that are generated by the contraction are transmitted to the skeletal system through the tendon, allowing joint movement. On the other side, ligaments connect bone to bone. They provide joint stability, and they prevent the dislocation of the joint. But one of the key but often overlooked structures within this system is the enthesis, which is a transitional tissue between where tendons or ligaments attach to bones. The enthesis contains 4 different regions that are the tendon part, the unmineralized fibrocartilage, and the bone. And it is essential for transmitting the forces that are generated by the soft tissue to the skeletal system. But despite being essential for force transmission, the enthesis is also one of the most vulnerable spots in the musculoskeletal system. And this vulnerability makes it particularly prone to injuries and disease. In fact, many debilitating orthopedic conditions involve the enthesis. And one of these is the rupture of tendons and ligaments. One clinically relevant example is the rupture of the rotator cuff tendon in the shoulder, which affects more or less the 60% of individuals over 60 years old, and its incidence is increasing also with aging. Autograft and allograft transplantation are the gold standard treatment for the ruptures of tendon and ligaments, but they present some limitation, leading to a high risk of fracture recurrence, chronic pain, and decreased muscle strength. But in addition to ruptures, also the inflammation of the enthesis is another common musculoskeletal disease. And tennis elbows and golfer elbows are classical examples of acute inflammation, which is a condition that generally resolves spontaneously. However, between the 30 and 50% of individuals that suffer from arthritis can develop a chronic form of inflammation. And this can lead to the formation of new bone tissue directly inside the tendon part of the enthesis, blocking the movement of the joint. This is also associated with chronic pain and can lead also to some degree of disability. But however, its wide incidence, the knowledge at the moment on the disease and the treatment options are very limited. Therefore, we think that developing representative in vitro model of the enthesis is crucial for advancing effective treatment for enthesis-related pathologies, improving also the soft to hard tissue integration, functionality, and long-term clinical outcome. Therefore, in Chapter 1, we provide a general overview of the current strategies that have been used for engineering the enthesis, and we explore also the challenges that are associated with tendon and ligament ruptures, and we describe also the need for developing representative in vitro model of the enthesis. In this case, miniaturized in vitro models are models that are designed to fit in a culture dish, and they provide a control environment for studying the cellular and molecular mechanisms that are involved in tissue development, injury, and repair. For establishing a miniaturized model of the enthesis, it's essential to replicate its structure, since it can help the cells to keep their function and shape. For example, in tendons and ligaments, collagen fibers are naturally aligned, helping tenocytes, which are the tendon cells, to keep their elongated shape and functionality. However, in traditional in vitro model, tenocytes tend to lose their proper shape and functionality, leading to the differentiation, which can affect also the clinical relevance of the model. Therefore, in Chapter 2, we develop a microfluidic piggyback platform that consists in a very small device in which we can control the flow. And by controlling the flow inside the device, we were able to generate collagen patterns with different degrees of orientation. We introduced in the chamber different micropillars with different shapes and spacing in between them. And in this way, we established a library of collagen patterns with different degrees of orientation. We then selected two patterns, here refer as circle 50 and rhombus 50, which show higher degree of alignment as compared to randomly aligned collagen fibers, here referred as blank, and we demonstrate that aligned collagen pattern can support the maintenance of an elongated shape in primary rat tenocytes, as compared to the blank control or as compared to standard tissue culture plastic. In addition, we also observe an increase in the expression of different tenogenic markers, such as Clearaxis and tenascin C, as compared to randomly aligned fibers or standard tissue culture plastic, and this was also associated with an increase in the expression of different tenogenic markers at the gene level after 3 days of culture as compared to standard tissue culture plastic, suggesting that aligned collagen pattern can support the maintenance of the phenotypes in primary rat tenocytes. In conclusion, in Chapter 2, we establish a microfluidic platform to control the alignment of collagen fibers. And we believe that the integration of this platform into tendon research can help the design of materials-driven strategies for tendon regeneration. 2D culture models, however, they do not accurately reflect the complexity and the physiological interactions between cells and their microenvironment. Indeed, in native tissues, cells are surrounded by a dynamic extracellular matrix, which provide mechanical support and the biochemical cues that are necessary to regulate cell fate and functions. 2D culture models, however, fail to replicate this complexity, limiting the interactions between the cell and the matrix. However, 2D culture models offer the possibility of micropatterning, which is anyway confined to one side of the cells, basically anchoring the cell to the bottom of the culture dish and introducing artificial polarization to the system. 3D culture models, such as spheroids and organoids, can increase the biomimicry, but they lack in the control over the environmental cues that are presented to the cells. Therefore, in Chapter 3, we introduce the floor-ceiling chip, which consists in two 2D opposing substrates with micropattern adhesive island, where we can control the spatial organization of the cell. In this condition, both the dorsal and the ventral side of the cells are in contact with components of the extracellular matrix, therefore removing the artificial polarization and establishing a pseudo-3D model. We believe also that patterning in 3D is relevant for connective tissue, such as tendon or ligaments, where cells are aligned and packed in between collagen fibers. We also demonstrate that with the floor-ceiling chip, we can control the alignment of the cells in 3D. By introducing collagen patterns in a line shape in both the top and bottom compartment of the floor-ceiling chip, we were able to control cell organization. By designing collagen patterns in a square shape, we were also able to modulate cell morphology. When the patterns were aligned, human mesenchymal stroma cells assumed a columnar shape, bridging between the top and the bottom compartment of the floor-ceiling chip. When the patterns were offset, cells were inclined, always bridging between the two different compartments of the floor-ceiling chip. So in conclusion, with the floor-ceiling chip, we were able to introduce micropatterning in 3D, and despite its confined structure, we demonstrated that the floor-ceiling chip is compatible also with long-term cultures, which are essential for studying different biological processes. Then, as I mentioned previously, the inflammation of the enthesis is a wide disease that affects many people worldwide, and the current treatment options are very limited to date. also because of the lack of reliable in vitro model where we can study the disease and test new therapies. Therefore, in Chapter 4, we introduce an enthesitis on-chip model that consists in a very small device that contains two chambers that are divided by a thin porous membrane. One compartment represents the tendon region of the enthesis, while the other compartment the fibrocartilage region of the enthesis. And after establishing an healthy model, we introduce inflammation in the system by adding an inflammatory cocktail that contains interleukin-17, interleukin-23, and TNF-alpha. In this case, we perfuse this inflammatory cocktail for 3 days in order to mimic acute inflammation and for 21 days to replicate chronic inflammation. After 21 days, we observe a general increase in the inflammatory response in the stimulated condition as compared to the unstimulated one, where we didn't add an inflammatory compounds. And this was marked by an increase in the expression for the receptor of interleukin-17, not only in the fibrocartilage compartment of the chip, but also in the tendon region, suggesting a possible crosstalk between these two types of tissues. In addition, after 21 days of inflammation, we also observe mineral deposition, here indicated in green, in both the fibrocartilage region and the tendon region. And this was associated with a general increase in the expression of different osteogenic markers at the gene level, suggesting a possible remodeling of the cells towards an osteogenic phenotype. After 21 days of inflammation, we also perfuse celecoxib in the system, which is a widely prescribed non-steroidal anti-inflammatory drug, and we observe a general mitigation of the inflammatory response independently from the presence of the inflammatory compounds in the media. So in conclusion, in Chapter 4, we establish an enthesitis on-chip model, and we think that in future this platform could be used for drug screening application, personalized medicine, and also the development of target treatments for enthesitis. Moving on Chapter 5, we then introduce a 3D spheroidal model of the enthesis that is based on the ability of spheroids, which are cell aggregate, to self-assemble and interact. Our approach is based on the fusion of ligament spheroids and osteogenic spheroids derived from human mesenchymal stroma cells. And for the fusion, we basically seed and culture the two different types of spheroids inside the same well. Indeed, we use microthermoformed microwells with an hemispherical shape, which allow us to control the spheroids formation and also monitor the fusion process over time. We generate ligament spheroids and osteogenic spheroids with different levels of maturation, and upon fusion, we obtain 4 different combinations according to the level of maturation of the single aggregates. After 10 days of fusion, we observe the formation of a core shell structure, where the ligament spheroids, here indicated in red, surround completely the osteogenic spheroids, indicated in green. We also observe the deposition of collagen 10, which is one of the main components of the extracellular matrix of the enthesis, suggesting that from the culture of ligament spheroids and osteogenic spheroids, we were able to form a third tissue phase. This was then confirmed by gene expression analysis, where we observe an increase in the expression of different markers that are related with enthesis maturation and formation. Finally, we also describe the scalability of this approach in forming larger tissue up to the millimeter scale, where we can still detect the expression of markers of the extracellular matrix of ligament, bone, and the enthesis. So in conclusion, in Chapter 5, we use spheroid fusion as an approach to recreate a complex ligament-to-bone interface, where we were able to detect some of the components that are found in the native enthesis. So to summarize, the miniaturized model that we propose are designed to recapitulate key physiological and structural features of the enthesis. We start with engineering the substrates in order to increase the biomimicry of the system. We then introduce three-dimensionality into on-chip platform in order to recreate a tissue with an architecture that is similar to what is found in the enthesis. We also use organ-on-chip technologies in order to recreate a pathological model of the enthesis. And then, through spheroids manipulation, we were able to obtain microtissue that have the same characteristic of the ligament-to-bone interface. So in conclusion, we think that the model that we propose can give the foundation for the development of physiological relevant enthesis model, with broad application, which can range from fundamental biology to drug discovery and regenerative medicine. And with this, I want to thank my supervisors, the collaborators, the founding agents, in particular the Materials-Driven Regeneration Program. Thank you for your attention, and I will give the word back to the prorector.
Thank you very much, dear candidate, for a clear presentation. Now we start the opposition. We start with the chair of your assessment committee, that's Professor Honing, and he's professor in analytical sciences at Maastricht Multimodal Molecular Imaging Institute. The floor is yours.
Thank you, Mr. Prorector. Dear candidate, congratulations with, I would say, this thorough and challenging work you presented here. As a non-specialist, very interesting to see what cells are doing and sometimes not doing. But of course, I have a lot of questions about this. And a little bit on how we should position cell-based and in vitro-based assays into what it is really telling us. And maybe start with page 236. There you conclude that the slow adoption of microfluidics is due to conceptual but a lot of practical challenges. You describe what you have been doing, but also in your presentation, what are the practical challenges then?
Well, highly esteemed opponent, thank you very much for your kind words and question. Regarding the challenges, the first one that comes to my mind is to keep the cells alive inside the microfluidic device.
Yeah, because you said sometimes they die for at least 20%.
Yes, exactly. One of the big problems that can be related to this is also the formation of bubbles inside the microfluidic device that block the perfusion of media, so the perfusion of nutrient to the cells. So it's important in this case to have control on the system that we are creating in order to avoid first bubble formation and the second to create a physiological environment for the cells to survive and proliferate and differentiate and also do their function.
Because I saw also, which is logic, that when you change the matrix or the scaffold, how you call it the scaffold, you get other cellular response and they will more die or not die or do what they do or not.
Yeah. Yes, exactly. Also, this is something that we need to optimize for every type of cells and also sometimes also for every type of design that we use. Because also sometimes we want to keep cells inside a specific channel. So in this case, we need to use also matrices or hydrogel in order to form like this kind of barriers also between the different channels. And this allows us to perfuse specific type of media also in the different compartment of the chip that are necessary for the cells.
So then from this, I would like to go to proposition number 3. So if one of the paronyms can read proposition number 3.
Microfluidic platforms enable the creation of physiologically relevant compartmentalized microenvironments, allowing for the controlled differentiation of tissue-specific cell types and the study of tissue interactions.
Thank you very much. So now I'm going to challenge you a little bit because you said it's still challenging. You explain me that, but you also dare to say in this proposition that you were able to make physiologically relevant conditions. And then I'm still wondering because that means you cannot do it, but you do it. So what is relevant in the sense, what parameters makes it relevant for you?
Well, I think in that case, for example, for the enthesitis and chip model, for us the relevant part was to obtain the different cell types that are found in the enthesis. In this case, it's very limited. Well, it's a bit limited, the model, because we just focus on the tendon part and the fibrocartilage part. But already to see the differentiation of the cells and the expression of specific markers that are found in the two different types of tissues we can somehow go closer to the physiological model of the tissue.
That means the real function of the cell system compared to real life.
Exactly. And also I think for that type of chapter, also when we saw mineral deposition after inducing inflammation, also that was for us like an indication that the model that we created is similar to what we have in physiological conditions.
But later, just to interrupt you, now the interesting thing is that many of the properties of the cell system and the tendon and alignment is on basic of forces, shear forces and so on. And in constructing them, you cannot use those forces. So I was searching a little bit on what is relevant here and how relevant your model really is in terms of physics.
Yes, I think if we want to recreate really a relevant model, we should include also the mechanical forces in this case that we didn't have in our chip because for practical reason, because it's quite challenging to recreate also the mechanical forces that are present in the enthesis because we don't have only stretching but also compression. So the part of the tenocytes are more exposed to stretching while the fibrocartilage and the bone region are exposed only to compression.
Clear. Now we go to a cellular level because when I look into the discussion starting in 212, that's actually the discussion of your thesis, you say actually, when I build this system, there is about, I calculate them, 8 or 9 parameters, cell-cell interaction, cell adhesion, who play a role. And then I think, that's extremely complex, especially cell-cell communication. And then I went to especially chapter number 5, and in page number 5 you explain how you make this co-culture and how you look to the biological effect, and I suppose that biological you mean chemistry. I'm a chemist, so maybe
exactly.
It's not biological, it's all chemistry. But the interesting thing is you explain that in the extracellular matrix there is collagen 1 or 2, depending on the days. And then I look to this other 2 compounds, decosin and tenacin. And when I look to the molecular structure of these molecules, these are extremely large, complex glycoproteins, apart from the physics there. But I see also their kinetics in terms of release is completely different. So how, and do you have an idea how you can use this data to understand how this alignment and this collagen forming of the cells really occurs?
Yeah, so these are also components of the extracellular matrix of the cells, so in this case of the tendon. And generally they are secreted and they are released in response also to mechanical stimulation. So what is happening is that when the cells are stretched, there is a different mechano pathway that are activated.
Yeah, but just to go further, when I look to them, so the kinetics of release between 1, 7 and 21 days is not linear, and the collagens go first, it seems, and then suddenly this other complex glycan protein. So what does it tell us?
Yeah, so what we assumed this was the level of maturation of the spheroids, because especially for decorin, it was already reported that there are different amounts of decorin according to the maturation of the single spheroids.
Oh, there's polymorphism in the type of proteins, so it's not one defined molecule.
No, it's not only one. We have a combination of them, and by this combination we can estimate the level of maturation of the tissue that we are
last question
studying.
as an analytical chemist. I was looking how you analyze them or quantify them, and I did not see nothing in the section on description of the method, or I missed it. So what were the assays? How did you calculate the quantity of these compounds in the extracellular matrix?
So we quantify the immunostaining, so we quantify the amount of area of the spheroids that was covered.
Oh, but it was semi-quantitative
exactly,
then. Okay,
exactly.
Then I'm happy. Then
yes.
Thank you very much for your answers.
Thank you, Professor Honing, for your position. Then we come to the following opponent who's online, and that is Dr. Michela. Her expertise is in vitro models of disease in the Institute of the Department of Pharmacology in Utrecht. And thank you for coming all the way from Utrecht, albeit online, because the University of Maastricht is always glad to have guests from outside. The floor is yours.
Dear candidate, congratulations on this very impressive piece of work. I learned a lot, and together we are going to dive deep into several aspects of your thesis, and probably you are going to enlighten me with some of the questions I have. I would also like to extend my congratulations to the supervisory team. I'm sure that they're very proud of you and your work. So let's dive into it. I learned about enthesis. And what I learned is that it's a complex multi-tissue interface. So my question for you to, let's say, to an icebreaker, how would you define a functional enthesis model in the context of in vitro modeling?
Yes, esteemed opponent, thank you very much for your kind words and for your question. Well, functional model of the enthesis I would include something that can respond also to the mechanical stimulation, because at the end, this is the real function of the enthesis, so to transmit the forces that are generated by the soft tissue to the skeletal system. Apart of this, I think also recreating the different zonal architectures of the enthesis, including the cell types and also the matrix component, is essential for establishing, in this case, an in vitro model of enthesis.
So in your thesis, you present several models. Are they models of functional enthesis? Are they pseudo models? Can you comment on this?
So,
based on what you just mentioned?
That we establish, I think they are simplified model of the enthesis, that they can replicate some of the structure and some of the key aspect of the enthesis, but not the entire complexity of it. And for this, the reason why we decide to focus also on some part of the enthesis is because the tissue itself is very complex, so containing different type of cells, different component of the matrix, and so we tried to go step by step. And then the model that we have can be also the foundation to then increase the complexity of the model itself.
So with this answer, I would like to ask one of the panelists if they could read the proposition number 4.
By fusing two distinct tissue types, a transitional third tissue phase can form.
So, chapter 5, I want to discuss about this transitional third tissue. So in this chapter, you combined MSCs with fibroblasts to form an enthesis-like zone. Yet you don't show, or I maybe didn't see it, or I didn't grasp it from your thesis, a spatial ECM gradient or zonal markers or some transition phase. And we can have a look a little bit at the TEM image. Let me see if I can spot it. I cannot find it now. You have a TEM image with your spheroids, so the difference between the two of them. Do you see any zonation being formed?
So
or they are just two. Yes.
What we observe is that so from the fusion itself, like the ligament spheroids completely surround the osteogenic spheroids. So in this case, we have one region in the aggregates that is mainly composed of the ligament part and another one that is formed from the bone part. In between, we saw the deposition of collagen 10. So in this case, we have like a kind of zonal organization because we have like one region only that is around the aggregate with the ligament part and one region that is more inside with the lindon, with the bone part. So in this case, we have a semi-organized structure, I would say, with a zonal also organization of the matrix of the different type of tissues.
And do you think we should investigate more in having more collagen and more aligned collagen? And how would you see this using this model?
Definitely. So I think there are actually many things that we could do. For example, to characterize better this model, we could do also special analysis on this to really identify how the markers are distributed in the different part of the tissues. Regarding the collagen alignment as well, we could see if the collagen fibers are also organizing in a proper manner, so to have a more organized part of collagen in the outside part of the spheroids and more disorganized mainly in the inside part of the aggregate. But yeah, for sure.
So I think chapter 5, we can move on. And probably not surprisingly, I'm going to ask you something about the pharmacological validation of your model. And you have used celecoxib to reduce inflammatory markers. But what does this actually tell us about your model and the value of your model for in a pharmacological context? Could this effect that you have shown have occurred in any generic inflammatory tissue model? And what makes your model unique, predictive, informative?
Yes, so the reason why we use chelecoxib is that it was already used in another on-chip model of osteoarthritis, and so we wanted to show the compatibility of our model with drug screening application as well. So we decided to use with a drug that is widely used also on on-chip platform, and just to test if we could actually able to see like a response in the inflammatory response of the cells after the introduction of the drug. Of course, it's not the best. In the clinical setting, is used also in combination with other drugs that are trying to reduce the inflammatory response, especially in the case of enthesitis. And for example, for enthesitis, is used also in combination with apremilast, that is another anti-inflammatory drug that is more efficient in the treatment of enthesitis. So I think it was not the best choice, but it was easy to use because it was already reported like in on-chip model for model of osteoarthritis. So like an in vitro model that is quite similar to the one of the one that we established for the enthesis inflammation.
And when you have such a complex cellular system, how do you know, for instance, where the inflammation is coming from? Because if you are going to stimulate the cells separately, you might have a response, but when they are together in a complex system, they might have a different response. So my question is for you, how should we normalize, for instance, this kind of data when we have multicellular systems?
Yeah, that's a good point. So for the enthesis inflammation, we know that inflammation is starting from the bone part, so from the mesenchymal population, and is then slowly migrating towards more the tendon part of the enthesis. How we can correlate this with our model, it's a bit complicated also because at the moment we really lack of animal models or in vitro data that can actually describe this process in vivo. I think that what we tried to do was to mimic also these waves of inflammation that is moving from the bone side more towards the tendon part of the enthesis and of the model that we established. But yeah, to get actually this translation is a bit challenging right now.
I appreciate the honesty of the answer, and I agree with you. And with this, I will hand it over back to the rector.
Short question and a very short answer. Well, you give back the word. Sorry, I thought...
Yes.
Understood. I thought you were asking me if there was time, but then I propose that we proceed. Thank you, Dr. Michela, for your opposition. Now we come to the following opponent, who's also a member of the assessment committee, and that's Professor Bouten. Her expertise is cell matrix interactions in cardiovascular regeneration in the Department of Biomedical Engineering in the University of Eindhoven. She's online, and thank you very much, Professor Bouten, for coming all the way from Eindhoven, albeit online.
Thank you, Mr. Pro-Rector. Dear candidate, my apologies for joining online. I would like to congratulate you with this nice work and also for your beautiful contributions to our consortium, the Material-Driven Regeneration Program. I also congratulate your supervisors. I would like to start at the end of your thesis. I have two main questions, one about the ultrasound stimulation and the other one about regeneration. I would like to start with the last part, which is also your last experimental chapter. And the other components refer to it, but they didn't actually mention it. I want to ask you first, in that chapter, the last chapter, you used ultrasound as a mechanical stimulation. My first question, what was the motivation to pick ultrasounds? Did you want to align something? Did you want to evoke a chemical response, trigger a mechanical stimulation pathway? What was your reason to do this?
Yes, highly esteemed opponent, thank you very much for your questions and also for your kind words. So the reason why we use mechanical stimulation was that we wanted to activate some mechanotransductive pathway that then we didn't have the time also to analyze, but we wanted to expose the cells to a mechanical stimulation in a way that the cells were not embedded in a gel, because we want them to remodel by themselves without any imposition like from the external microenvironment. And so this is the reason also why we use acoustic stimulation for the stimulation, for the mechanical stimulation.
Okay, so you wanted to have a remote trigger without interfering with the tissue. Now, there are several ways to choose the right trigger. You build a bioreactor, or you used a bioreactor. Can you tell a little bit more about this bioreactor? What did you design it such that the acoustic wave was correct, or could you position your tissue compared to the acoustic wave?
Yes, so the bioreactor contains basically a chamber that is in PMMA, and we make a kind of chambers inside to hold the microwells where the spheroids were cultured, and then we use two commercial sound sources that were bonded on the lid of the well plate to generate basically the sound wave. And then also we calculate the distance between the sound source and where the spheroids were cultured to see. We compare also to previous model to see if the cells were actually able to sense the acoustic stimulation that we were imposing. And this was based also on previous studies and previous publication also in our institute.
Yeah, I've seen that publication. So could you tell me, because if you apply an acoustic wave, there's also reflection, right? And the wave comes back. And of course, this is a very complex situation. Do you know exactly what the cells feel?
Yes,
in your situation?
Yes, that's very true.
Study.
Yes, we don't know exactly what is happening to the cells. And also, I would say also in which depth the acoustic stimulation are interfering with the spheroids, because we know that when we are exposing tissues or cells to acoustic stimulation, they can penetrate up to 300 micrometers, more or less, in the aggregates. The one that we have, they are smaller, so we think that actually they can sense the stimulation that we are perfusing to the cells. But then when we analyze the data from the acoustic stimulation, we see that there is an increase in cell proliferation and also matrix deposition. And so we think that actually the acoustic stimulation is right for this type of cells because they are responding also in a similar way to what is happening in vivo. Because acoustic stimulation is used also for the treatment of enthesis inflammation or osteoarthritis. And the main reason is that with this stimulation, cells are stimulated to proliferate and secrete matrix component. And so in this way, we could have like a similar results in our in vitro setting.
Yeah, that is clear. So you link it to the clinical application, which is a strong point. But it remains a black box, right? So there's some work to do there. What I usually see is there's a lot of calcium aggregation and mechanotransduction in those cells. Now, I would like to switch to the second part, and we should do that probably a bit quicker. There are other opponents. The title of your thesis, so I flip to the complete start of your thesis, is about advancing enthesis regeneration. Now, on a critical note, I've seen beautiful models, better insights, but I've not seen regeneration. So how would you translate your models towards, and there is something in your discussion, but it's more about optimizing your models. How would you translate your knowledge to regeneration and perhaps even material-driven regeneration?
Yes, so if we start, for example, from chapter 2, where we use the microfluidic platforms to align collagen fibers, in this case, from the interaction between the materials, that is the collagen and the cells, the tenocytes, we saw that we can improve the maintenance of the phenotype of cells. And so if this is applied to an in vivo settings, we can improve actually the phenotype of the cells, and this could help also the regenerative process later, because if cells are kept for a longer time in their proper shape and in their function, this can help also the regeneration of the tissue and the synthesis also of matrix component or other factors that help cells also to respond to damages. And then if we think, for example, on the fusion chapter, in that case, the regeneration, we can see it when we applied also mechanical stimulation, as we mentioned before, with the synthesis of new matrix. And this can lead also to the repair of the tissues, as is already demonstrated like in the literature, that this is essential also for having like a reparative property in the tissues. And then if we think on the other side of the inflammation chapter, so with the enthesitis on chip model, that model, I think it could have potential because it allows us to study the inflammatory process and eventually also how the tissue is responding to this. And so it could be used actually as a platform to study the regeneration after inducing inflammation or after inducing the damage in the tissue. So I think they have the potential to study regenerative processes, all of them, like also in a different extent.
So in hindsight then, because I agree with you, but yeah, it's a step up and using your models for regeneration. In hindsight, how would you adapt your title of your thesis? If there's no regeneration in your thesis, how would you adapt it? I'm curious.
Yes, maybe I will adapt it, advancing enthesis understanding, maybe through miniaturized models.
Yeah, I fully agree. We move from understanding to design, which is great. Thank you very much for your responses in our discussion. I give the word back to the pro-rector.
Bouten for your opposition. Now we come to the following opponent, who's also a member of your assessment committee, and that's Professor Balmayor. She's professor in experimental orthopedics and trauma surgery at the University of Maastricht and in the Department of Orthopedic Trauma Reconstructive Surgery in the University Hospital in Aachen, and that is in Germany. Professor Balmayor.
Thank you very much, Pro-Rector. Dear candidate, my congratulations also to your supervisor. I think I enjoyed very much your thesis, and I think it's important to highlight that this is indeed, in my opinion, a contribution to a field that is very poorly investigated. So I think these models will open many doors, and I just heard that we have a lot to do still. So I think this is a work in progress, but it's certainly very, very important. I will start with the first question, and it's more a general question. I'm completely sure that you are aware that entheses are not the same in all the different localizations, right? How do you think we could adapt your different investigated models to personalize them to use for rotator cuff, which you very well mentioned, patella, Achilles, other type of entheses?
Yes, esteemed opponent, thank you for your kind words and also for your question. Yes, we know that entheses are different according to the position in the body that we are considering. One thing that we could do actually is to isolate cells, for example, from the different type of the region in the body and to culture them inside a microfluidic chip. But we could actually also adapt the cells that we have in a way that they can recreate the different type of tissues. Because, for example, we know that the ratios of collagens are different according to the different part of the entheses that we are considering. So we could actually try to engineer this system with different types of collagen and different combinations or different amounts in order to recreate also the different type of entheses that we are actually considering, for example. Also, another thing that we have to consider is that they are exposed somehow to different types of mechanical stimulation, and this can lead also to a different matrix composition or different differentiation in the cells. So this is also something that we have to take into consideration and to recreate, for example, more uniaxial stretching or biaxial stretching if we are introducing the mechanical stimulation to the system to recreate also the different parts.
Fantastic, because that is exactly what we observe. You come from the mechanics and you see how the different entheses are indeed on the different mechanics and that affect the gene expression, the matrix deposition. So you beautifully summarize it all. Now, it's very interesting for me to see that you are so aware of the needs of mechanics in your system. However, we have a little bit of a lack of the mechanics in your development system. So what would be the challenge then? Because you say to us already, I will need to recreate the stretching, compression. I see that you see that in the shoulder, I need another mechanics that in the Achilles. What do you think will be the technical challenges to put in your system a little bit of mechanics?
Yes, if we consider like an on-chip model, for example, we should have different compartments that are exposed at the same time to different type of mechanical stimulation. And this, from a practical point of view, is quite challenging, I would say. Because, well, one thing that we actually could do is to recreate, for example, different organ-on-chip systems, so to have different type of chip that are isolated. We can expose one to stretching, for example, another one to compression, and then connect them through tubings in a way that the media that is perfusing one chamber can then reach the second one. So in this way, we can have like an exchange on the media and different factors that are released on the different types to recreate a more physiological system. But of course, I think the best way would be to have the same device in which we are applying the two different type of stimulation at the same time. And this, I think, it will be particularly challenging also to control the intensity of the stimulation and how much the cells can sense it and how much they are exposed actually to the stimulation itself.
Very good. And I hear a lot of complexity, and I like, because we like to make, as scientists, a high complex models for what is already a highly complex tissue interface. So I cannot stop but wondering, is that not very, very complex everything? When we use 2D co-culture systems or a 3D good biomaterial, as good biomaterialist scientists, and have a good in vitro system for enthesis regeneration, what do you think is the strengths of the tools that you are bringing to the table?
Well, I think with microfluidics, the strength that we have is the compartmentalization that we can offer to the system. So to have different type of tissues that can be exposed to different factors, different stimuli at the same time. Yes, the challenge part of this is actually to control all of this at the same time, no? And to have it like in a reliable way, in a way that is mimic actually the physiological interactions of the physiological effect of the tissue and how, and apply also the mechanical forces in a way that can mimic what is happening actually in vivo with the same intensity, same duration, and so on. So I think this would be like also the most challenging part.
Very good, very good. I have another question, because in chapter 5, you use ACL derived, right? Fibroblasts and MSCs. The MSC's choice is clear, right? You also explain it very well. When we will consider another enthesis or another ligament insertions, PCL, something else in the knee, what will be your decision in terms of cell sorts? Which combination will you select? Only 2, perhaps 3? Would you need 3 cultures in terms of?
Well, I think with 3 culture will be more realistic, more physiologically similar to what we have in the enthesis. So actually we could have different combination, because we could start, for example, from tendon cells, we could have chondrocytes or hypertrophic chondrocytes, and then bone cells. So in this way we could have all the different cell types that are found in the enthesis. And actually thinking about this, it would be nice also to start, for example, from IPS. So isolate, for example, the cells from one patient, reprogramming the cells towards iPSCs, and then differentiate iPSCs towards the different cell types that are found in the enthesis. So in this way, we could have like the complexity, because we have the different cell types, and at the same time, we can start from cells from one donor and to recreate a more personalized also system or a personalized model of the enthesis, I would say.
Very good. Perhaps very quick, the final question. We heard already a very nice discussion with another opponent about the acoustic stimulation, right? And you say very nicely, it does indeed increase the matrix deposition for the type of cell that you were stimulating. But if I change the type of cells, then perhaps I need another type of stimulation. And in figure 8 from your chapter 5, you have microfluidics, bioreactors. You do in your thesis go back and forth. Can you very briefly mention to us what is the different type of stimulus that I will then get with a bioreactor or a microfluidic, for instance?
Yes, with microfluidic, we could have, for example, shear stress. That is also something that we have to take in consideration if we want to create an in vitro model, for example, of enthesis. And this could be beneficial for tendon cells, but it's important in this case to control the intensity of the shear stress that we are applying, because it was already shown that it can induce osteogenic differentiation. So it should be done in a controlled way, in order to stimulate the cells that we have to produce the correct markers and not differentiate towards another type of cells. Another advantage in microfluidics is that we can apply mechanical stimulation by integrating also flexible membrane in the device. So for example, if we have a flexible PDMS membrane and we culture, for example, tenocytes on top, for example, chondrogenic cells embedded in a matrix on bottom, and then we try to apply compression to the bottom, maybe the membrane will bend, so the cells on top will sense more the stretching, and the one on the bottom, they are more compressed. So in this way, we can recreate also the two different types of stretching and mechanical stimulation that we can apply to the enthesis itself. But yeah, I would say that shear stress is also like something that we have to consider.
Thank you very much. I'm very satisfied with the answers, and I give back the word to the prorector.
Thank you very much for your position, Professor Balmayor. Then we come to the following opponent, and that's Dr. Peniche. His expertise is enthesis regeneration in the Merlin Institute for Technology-Inspired Regenerative Medicine in the University of Maastricht. And today he has also accepted the role of secretary of this committee, for which I'm very grateful. The floor is yours.
Thank you very much, dear Prorector. And first of all, of course, congratulate the candidate for an amazing piece of work that I have enjoyed tremendously reading since I am so into enthesis. This book has been very entertaining and very useful also for me, as you deal here with a lot of biofabrication that I haven't explored myself. So thank you for inviting me to be part of these days. And of course, congratulations also to the supervisors for such a nice piece of work. So I would like to start coming back with proposition number 4, which has been already read by your paranims. And in this proposition, you refer to chapter 5 a little bit. And you talk about the formation of this third tissue interface by fusing two phases, or two different tissues. And that made me think that in the introduction of your thesis, you actually describe the enthesis as a succession of 4 different phases. So you go from bone to mineralized fibrocartilage, non-mineralized fibrocartilage, and tendon. So my question here is, do you think that it's possible to meaningfully study enthesis by looking into only 3 phases? And if yes, then which of the 4 phases you will leave out, and why?
Yes, esteemed opponent, thank you very much for your kind words and for your question. That's very true. We actually obtain more or less 3 phases, I would say, not the original 4. But the reason why I think it could be still important to have this kind of model is that the third phase that we observe, it's forming only from the co-culture between the 2 different cell types. So it's really a process of cell-cell interaction or cell matrix interaction that leads to the differentiation of cells towards a third phenotype. Of course, if we want to recreate like a more physiological model, we should have all the 4 different regions of the enthesis because each of them has a specific function and contains specific cell types. But yeah, it's a bit limiting the fact that it was there are actually only 3, but it's a process that is forming by itself. And actually, I think that considering that we have collagen 10 in the matrix, and collagen 10 is one of the components that is present both in the fibrocartilage region of the enthesis and the mineralized fibrocartilage can give us an indication of this transition tissue that is forming in between. So I think it's a first step towards a more complex system.
Yeah, no, I totally agree with you, and I would have done the same in your case. And it's good actually that you mentioned collagen 10, because I was wondering, collagen 10 is a good marker for what you just explained now, but I was wondering, would it be possible as well to look into collagen 2 in the same model?
Yes, we actually did it. We observed also some deposition of collagen type 2, but this was a bit more disorganized as compared to collagen 10. And with collagen 10, we could clearly see this demarcation, so this difference between the bone that was not expressing collagen 10 and the kind of enthesis part that was expressing collagen 10. And for collagen 2, we actually observed some expression also in the ligament part of the spheroids. So that's the reason also why we didn't consider at the end collagen 2 as more a marker for the enthesis, since there was this overlap between the ligament part and then the enthesis region. While with collagen 10, we had like a clear signal that was in between the two different types of spheroids.
Yeah, perfect. Yeah, very good. Then I was wondering, because when looking at collagen 10 again and the overlapping with decorin, I noticed that most of the overlap, you saw it especially on the ligament part, using the ligament 7 and B15 and the ligament 21 and ligament 7 and B21 and ligament 21 and B15. So what got my attention here is that in these both scenarios, you have the less mature ligament spheroid, so the ligament 7, and in the other side, or the other construct that behaves similarly, has the more mature. So perhaps you can elaborate a little bit there. Do you know where is this collagen 10 coming from? Is coming from the ligaments, or is so? Yes. What do you think about it?
Yes, that's a very good question. So the reason why we try to measure this overlap between the collagen and the matrix component of the ligament of the bone was to get an idea of where it was coming from. But we actually don't have a clear response, to be honest, because there is a different behavior according to the different cell type. What we think is that anyway, there is from the co-culture itself, so from this interaction, we could have like the remodeling of the cells towards more an osteogenic, an hypertrophic chondrocyte phenotype. And it's actually already proved, and it has been shown, that when fibroblasts are in co-culture together with osteogenic cells, there could be this change in the phenotype, leading to the formation more an hypertrophic phenotype in the cells. So exactly from where it comes from, we cannot say it, but we have an indication that maybe are the fibroblasts that are differentiating towards more an osteogenic phenotype later.
Yeah, very good, very good. Thank you very much. I would like to address what for me particularly is the elephant in the room, which is the fact that your approach for using spheroids, I consider it to be extremely bold and very innovative, especially when it's applied to enthesis, since you have already very well described that the enthesis is this succession of phases. However, in your spheroid construct, you have more a cold share configuration, right? Which is basically the opposite to what you see in the nature. So considering these key differences between your model and the biological tissue, what would you say that is the most relevant advantage that your model brings to the table that the available alternatives are lacking?
Yes, so what I think is the main advantage is that in our model, we can really study cell-cell interaction and cell-matrix interaction because we didn't add any external factor. So we didn't add any scaffold or any materials that can somehow influence the organization and the matrix deposition in the cells. So I think this is mainly the big advantage of our system, that the cells are free to remodel and they can form the interaction that they maybe form also in the natural tissue. But of course, having the spheroid model is not the best type of choice, especially for ligament cells, I would say, because we have, for example, higher cell density than what we have in the natural tendon, and we have like also different organizations.
You may finish your answer if you want.
Yes, so what I think at the end is that it's not the best model, but it has some similarity to the in vivo tissue. And then, of course, from this, we can improve the model like in many different ways, I would say.
Thank you. Thank you very much.
Thank you, Dr. Peniche, for your opposition. Dear candidate, Ms. Giacomini, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. And I request that you and your company on site, so here present and online, await the results of our deliberations and our return in this room. And therefore I suspend the meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen this academic ceremony. Dear candidate, Ms. Giacomini, the degree present has assessed the quality of your thesis and the way you defended it, and in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Habibovic is authorized to confer upon you this academic distinction in accordance with Dutch university custom, and I invite her now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Francesca Giacomini, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Coming to the laudation. Dear Dr. Giacomini, dear Francesca, it is a great delight to congratulate you on your doctorate, which has been eagerly awaited and is more than deserved. It required qualities, of course, such as scientific talent, commitment and organizational skills, as well as resilience in these challenging times, which included a pandemic during your PhD project. It also required belief in yourself and perseverance over several years. You have proven all of this, and your doctorate is a recognition and reward for that. Let me take us back a few years to when it all began and guide us through the major stages of your PhD to properly acknowledge them. It was still in the early years of our Merlin Institute when a Dutch academic consortium, including Maastricht University as a consortium partner, secured funding for a large-scale 5+5-year program on materials-driven regeneration, abbreviated MDR, within the gravitation grant scheme of NWO, which is the Netherlands Organization for Scientific Research. The Maastricht team was led by your promoter, by Mala Bibovic, and by Clemens van Pieterswijk, and involved also your co-promoter, Stefan Gieselbrecht, and myself. All four researchers made, among others, significant contributions to the grant proposal program section on regenerating the soft tissue-to-bone interface under which you have been eventually employed. Like many successful Merlin PhD trajectories, also yours started at the latest with a call-up and brief introduction of new Merliners during the weekly Institute colloquium every Friday in our social kitchen area. For your first study then on microfluidically aligned collagen to maintain the phenotype of tenocytes in vitro, you developed a user-friendly, bottomless microfluidic chip that, when reversibly mounted in a culture dish and perfused with collagen solution, allowed the controlled micropattern formation and aligned the position of collagen fibers simply on the bottom of a dish. You know this because you did it. After removal of the piggyback device from the dish, where tenocytes, what we heard also cultured on the aligned fibrous collagen, exhibited a more elongated morphology and showed an increased expression of dendritic markers compared to controls. In this study, Francesca, you mastered new topics, such as, with the help of a proven support team, the application of microfluidics for the creation of chemically and structurally biomimetic cell interfaces. That's my interpretation. After intensive communication, which we had with the reviewers and editors in multiple submission rounds and the corresponding revisions, which probably made you and the core team of authors feel as if they were preparing the manuscript a second time, the rather extensive manuscript found a fitting home in a nice journal, Advancement Healthcare Materials of the publisher Wiley, and according to its citations, seems to be well received by the community. In your study on enthesitis on chip, you differentiated human mesenchymal stromal cells towards an enthesis model around the porous culture substrates in a microfluidic organ on chip and induced a physiological manner from the fibrocartilage side of the model to different extents of inflammation, which you were then able to show to propagate to the model's tendon side, and finally demonstrated the capacity of the model to respond to a non-steroidal anti-inflammatory drug, what we also discussed here. This study, I found, was very well designed and conducted and proved again your true mastery, I think, in seamlessly switching between microtechnology and fluidics on the one hand and cell and molecular biology on the other. Following a similar strong team performance in challenging review rounds, just as with the first paper, the even longer manuscript settled once again in Wiley's Advanced Healthcare Materials. Now, your third study on the so-called floor ceiling, or 2 × 2 equals pseudo 3D, together with Uren Delger or Urna Tufsindor, can serve as a prime example for the collaboration of two junior scientists in our field. Completely in a remarkably short period of time, the study explored how 3D cell morphology and organization can be approached by 2 opposing culture subsets provided with cell-adapted protein patterns. The culture system stands out, I think. You mentioned it also due to the long potential culture durations between the 2 subsets, which are in the form of permeable nanoporous thin ion-tracked membranes, as well as the good observability of the cells between the membranes. The preview, and that is our outlook, of aesthetically striking columnar shapes we saw of fluorescently labeled cells adhering to geometric collagen tiles, both on the floor and ceiling subset, leaves us curious about the possibility that could emerge once we are able to precisely align the bottom and top subsets to each other in a potential follow-up study. Now, expressed in mathematical terms, your study on the formation of enthesis-like multicellular microtissues from ligament and bone spheroids, this would probably be something like A plus C divided by 2 equals B. That is my way how I read it. Fusing HMSC-derived bony spheroids with spheroids from anterior cruciate ligament cells revealed at the interface the de novo formation of a region with cells and matrix resembling the phenotype and chemistry of the enthesis, respectively. While you were not, obviously, immediately convinced of the concentric organization of the multi-microtissue, your supervisor was not completely unhappy, as consequently there is no issue with changing orientation of the tissue interface between replicates during their microscopic observation or also mechanical simulation, what we discussed. Francesca, you were a PhD student, the kind one could only wish for, professionally, socially, and personally. One could rely on you to get the work done, and to get it done, you did it very well. With your always friendly and helpful manner, your positive attitude, you were and are an invaluable member of our team. Again, my congratulations on your significant accomplishment, substantial and meaningful thesis, and a convincing defense today. Now coming to thanking all the people, I would like to extend my congratulations and thanks to the other members of the supervisory team, Pamela and Stefan. Pamela, for despite your demanding job as a rector of this university, still making time to regularly discuss the progress of the PhD project and to work through the draft manuscripts on the weekends. At this point, I would also like to thank, sitting here in the audience, Dr. Tamashebi Begani for all her support. Thank you, Nilfar. Thanks a lot. I would like to thank our other internal collaborators, among others, Dr. Anand, Dr. Bayo Barata, Dr. Domingos Mota, Ms. Aishan Loches, Dr. Emans, Professor van Griensven, Dr. Lopez Iglesias, Professor Moroni, Dr. Ro, Mr. Song, and Dr. Vermeulen, and of course, all the Merliners. And our external academic collaborators with the MDR Consortium, among others, Professor de Boer. Thank you also to the chair and members of the Assessment and Defense Committee, Professor Honing, Dr. Mejaya, Professor Bauten, Professor Balmayor, and Dr. Penish Silva, for the critical reviewing of the thesis, writing the reports, preparing and joining these defense, and for the interesting scientific discussions during the same. And also the pro-rector of the defense, Professor Nijhaus, for chairing it. I would also like to thank the Paranymphs, the Beetle, and the technical support team here at the Minna Brudersberg, and all people who have been following this event on-site and online. Last but not least, thank you to the main funders of our project, NWO, in the province of Limburg. And I would, of course, like to congratulate Francesca's family and partner, and thank you for your support. She did obviously very well, and you have every reason to be proud of her. We are happy and grateful to have you still with us as more a senior researcher now with the second phase of the MDR program. And as a team, we are really looking forward to the things still to come. I'm closing by wishing you wonderful celebrations with your loved ones ahead. Enjoy your success. And with that, I now hand the floor back to the prorector.
Thank you, Professor Truckenmüller, for a beautiful laudation. Dear Dr. Giacomini, also on behalf of the University of Maastricht, I would like to congratulate you with the degree you have acquired. I hope you can celebrate it in the right way. This is probably one of the most important days in your scientific career. And I extend my congratulations, of course, as Professor Truckenmüller also said, but now on behalf of the university, to your parents, of course, but also to your supervisors, Professor Habibovic, Professor Truckenmüller, Dr. Kieselbrecht, and of course to your partner, family, friends, and colleagues on site and online. I would also like to thank the members of the opposition for all the work, and of course the Padel and the technical support team who made this hybrid ceremony possible. I come to a closure of this ceremony, but I have two announcements. We are going to stay here for a while to make a photo with the two external opponents on the background. So I ask you, the audience, to already go to the rafter where the reception is. And the photographer can also, of course, stay here. Then we are going to make a picture at the foot of the stairs. And we can do that also with your partner and parents or family, what you want. And afterwards, we go to the rafter. And if you think, when can we congratulate the young doctor? It's very simple. You follow us at a certain stage, we go first, and then you come. All right, having said it all, I now close this ceremony.
Just take that one for the picture moment.